Okay, hi everybody. Welcome to Q&A with A&V. I'm Vincent Racaniello, and with me is Amy Rosenfeld. We're both professors at Columbia University. Hi, Amy.
Hi, Vincent.
It's 8 p.m. here in the Northeast, and we have 30 minutes or so to answer your questions about SARS-CoV-2 and COVID-19. And then Amy has to go do an experiment.
Yes, we have to add overlay to plaque assays. Make new plates. Got to progress.
So one question here from U.S. Once partial immunity is established, we will have another common cold coronavirus to update immunity with minor sniffles every December, right? Well, that is the hope, right?
It's a hope.
Becomes another common cold coronavirus. But Amy, you think it may take more than a few years, right?
Yes. There's always a naive population. So,
Ralph Barrick, who is the expert on coronaviruses, says he thinks that's what's going to happen.
5 years or longer?
Well, he didn't put a timeline on it, so.
Yeah, I agree with him, but it's not 5 years. Plus, you always have the people who are immune compromised and who won't take the vaccine.
Well, those people actually get sick from common cold coronaviruses, right?
Yes, but there are also people who won't take the vaccine.
Sofet writes, hey, do you believe SARS-CoV-2 has made its way into wildlife populations?
It's in deer mice.
So Tony Shounce over at Colorado State is worried about that. Deer mice are highly susceptible to infection. So since people have contact with deer mice and researchers go out in the field, to study deer mice. So it's very possible that the virus will get into them. It certainly gets into other animals like mink, right? So I think a big worry, right, Amy?
Yeah.
Troy got a message from Moderna saying they're going to notify us if we got the placebo. What are the implications of unmasking their study at this point? That's very interesting, don't you think? They're going to tell people if they're placebo group?
Seriously?
Well, that's what Troy said. He got a text message. So anyway, if that's the case, they have to give all the placebo people the vaccine. It's part of the agreement, I'm pretty sure.
I understand that, but how are you going to have the control to make the comparison for later time points?
That really throws the study out the door, doesn't it?
Yeah, I'm not sure. I don't agree with that.
So that's a big issue. To break the code right now, then you really lose your data. But that's been done before. If people feel that this vaccine is so good and that we need to give it to people, they'll just break the code and move forward.
Yeah, I'm not going with that.
Randy wants to know, why is the vaccine creating symptoms COVID? If the vaccine doesn't contain the weakened virus, just mRNA?
Well, the symptoms are half cytokines.
Pardon me?
They're half dependent on cytokines. We know, like from rhino and this, probably that by the time virus peaks, our symptoms peak, virus peak has passed. So it's all the cytokines.
Well, the mRNAs, of course, will make a viral protein that's foreign, and you'll make an immune response, and that'll be protective.
Yeah, but you're still going to stimulate something.
What, when you inject the vaccine? Is that what you're saying?
Yeah.
That's why some people who get it will feel, soreness in the arm because there's an adjuvant in it, some fever, some muscle aches, and so forth.
Yeah, I think I saw 15% of Pfizer's record side effects like that.
All right. I was wondering, 3ND4K51 wants to know if SARS-CoV-2 vaccines would provide some protection against other coronas, including common cold and yet undiscovered coronaviruses.
Depends on how homologous the spikes are, right? Probably not to the common cold. We don't see any. I really highly doubt that we're able to predict what will be in the future. You think we're outsmarting Mother Nature? No.
I don't think these are going to be cross-reactive, though. They're really designed specifically for SARS-CoV-2.
Yeah, they're definitely not going to be cross-reactive.
Lisa wants to know, what happens if these vaccines aren't kept at the extremely low temps they require? And how can the end user be sure they've been kept at the proper temperature during their transport? This is a good question. So, you know, the Moderna and Pfizer vaccines have different requirements for maintaining stability. You know, Moderna can last at refrigerator temp for 6 days or something like that. I don't remember the number.
Yeah, something like that.
Pfizer's has to be kept at minus 70 C until it's open.
No, I think they're changing it.
They think so. But anyway, if you don't keep them at the right temperatures, they lose potency. And so you won't get a full dose, essentially, the longer it sits at the wrong temperature. And it's very hard to know that, right?
Yeah, but do you question when you go to the grocery store and you buy a gallon of milk that was transported at the wrong temperature?
Could have been, right?
It could have been, sure. But you don't think about it, right?
No, but this is different. This is not milk. This is your life, right?
The same analogy holds.
So I think that...
you know, the company is doing it themselves. Like, they don't want another cutter incident. So I highly doubt that there would be compromising.
Paulo wants to know how many particles are needed for infection in an animal model. Is one enough? And does this depend on each organism?
Definitely depends on each organism.
So I have seen some experiments in animal models. I think the Barrick, you know, the mouse model with the passage
10 amino acids,
like 100 PFU is enough to infect those mice. But one, I think, is not enough. One is not enough.
No, but it all depends on the model, right?
Depends on the animal.
It's not extrapolatable to non-human primates.
Kelly wants to know, as far as vaccines go, which type would be the most effective for middle-aged to elderly people? Well, we don't know because we've only tested, we have results for just 3 so far, right? Moderna, Pfizer, and AstraZeneca. And those have been tested in middle-aged to elderly people, and they're looking good, as good as in other age groups. So I'm elderly. I would take any one of those so far. What are the chances of vaccine mRNA finding its way into cardiomyocytes or other tissues? What do you think of that?
I don't think that there's a lot of high chance. I mean, it's a small amount. Your body's huge volume, and it's going into a solid muscle.
It's a good question. We talked about that on the cardiomyocyte episode, remember?
Yes.
You were on that episode, in fact.
I know. We talked about T-tubules and cardiograms and making Ian a guinea pig.
So if the mRNA got to the cardiomyocyte, you worry that it might induce fusion, right?
Well, your response was that these are all encoding the fixed pre-fusion form of spike.
Perfect. You remembered. That's great. Yeah, these are pre-fusion forms, so they're not going to cause fusion. That's why I have Amy here. She remembers everything. Did I say in a previous episode about the vaccine breaking down rapidly after injection? The mRNA.
Why would it break down rapidly?
No, they're designed to be stable and, you know, and get taken up into cells. So I think their fate is to be taken up into cells, you know, mainly antigen-presenting cells and so forth.
Well, anything that has a lipid membrane.
How do these nanoparticles cause an immune reaction? Well, the nanoparticles themselves are adjuvants. They act to stimulate innate responses, which helps you get a better antibody response. And then, of course, the particles are taken into cells. The mRNA comes out, and it's translated in the cell to make the protein. And then the protein is recognized as foreign and
digested up and put on MHC.
That's right. I traveled to Korea. I have traveled to China, Korea, and came back on 12/5, got sick the next day, one of the worst flu-like symptoms. I had flu in October. Antibody test was negative in May. Could it have been COVID? Sure.
Yeah.
Absolutely. I think the earliest known case in China reported was 12/1. So sure.
No, it's November 17th.
Well, that's not in the journal articles. That's in some Wall Street Journal thing that you read, right?
No, it's in the Lancet.
So say the difference again between SARS-CoV-2 and COVID-19.
SARS is the virus and COVID-19 is the disease.
Yeah, SARS is the virus and COVID is the disease. Is the virus really mutating, or do we just not know enough yet? Can we make a big difference by changing our relationships with nature? Well, yes, all viruses mutate every time they reproduce. And SARS-CoV-2 is no exception. There are changes in the genomes that occur.
Yeah, but most of them are unimportant. They're silent.
And Amy is saying most of the changes are unimportant and very few make a difference. In fact, many you don't even see because they inactivate the virus. So far, there's one strain and they all seem to be the same. Sue wants to know if they are not allowing family members to visit relatives in the hospital after 10 days after the onset of symptoms. I asked Daniel Griffin this, and he said it depends. If you're really sick and it looks like you're not going to make it, they may let you go see your loved one. He said, or if the patient gets 2 PCRs over a day apart and they're negative, you could go in as well. And Lila says, love you, Amy.
Yep.
You got fans, I told you. Hey.
Let's not get carried away.
I hear the vaccine contains SARS HIV RNA plus HIV RNA. No, absolutely not.
It's a plasmid that's transcribed into RNA, and the RNA is then put into a particle. It's ridiculous.
Here's a good question. Are our own extracellular vesicles as short-lived as the mRNA vaccine's lipid nanoparticles?
No.
They're longer-lived?
Yeah, some of them.
Some of them are longer-lived, Amy says.
Depends on what you're assaying for.
Yeah, it depends what you're assaying for. Leisurely asks, if T-cell response was present in 40 to 60% of blood samples prior to COVID, would that affect the results of vaccine development?
Why?
You're saying virus-specific T-cell responses?
I would assume that's what she meant.
Yeah, I don't know. It's a good question.
Why would they affect vaccine development? Most of the vaccines are measuring not the presence of neutralizing antibodies.
Yeah. Rush the vaccine? Oh, no. My teacher was asking me about the whole peer process for vaccines look like because she was worried about it being rushed. So here's the story. It is fast, right? It's fastest in history, but I've been watching it since the start, and everything is being done well, at least by Pfizer and Moderna. AstraZeneca, they had some issues there. But all the safety results look good so far. Phase 1, Phase 2, and now Phase 3. And, in the end, it's a matter of risk. What risk do you want to take? Do you want to have the risk of getting serious COVID, or do you want to have the risk of a minor side effect? So that's what you have to decide, right? Hello, Amy, you back?
I am here.
You hear echo?
I still hear echo.
If you had a choice, Sharon wants to know, if you had a choice, what vaccine would you get?
Out of all of the potential ones, or the 3 that are available?
That's the question.
Well, it's vague, so I don't know what it means exactly.
Well, I would say of the ones that are looking good so far, well, there's 2 mRNA vaccines and an adenovirus vaccine. If I had a choice, I'd take the mRNA vaccine out of those.
Yeah, I would, but I might hold out for an attenuated infectious virus-based vaccine.
Yeah, Amy says she would hold out for an attenuated virus vaccine. All right, Edwin writes, have any of the vaccine manufacturers reported whether people that have been vaccinated and then infected with SARS-CoV-2 still shed virus? That's a great question. They have not. As you may know, the primary endpoint—
they're not looking—
is disease, prevent disease. Some of the companies are taking nasal swabs, and later on they'll look to see if they get infected and shed. So maybe at some point we will know, but not now.
Well, Well, just based off of the non-human primate models, they're not preventing transmission.
Is there any reason to think that the vaccines are giving an early immune boost that will wane? The live virus vaccines might provide a temporary non-specific boost versus the mRNA. So I don't know. That's a great question. Will this protection wane? We have no idea how long it's going to last. We just have to try it. But all we know is that a few weeks after the last boost, what was it, 10 days or 14 days, Amy? You get 90.
That was 7.
7 days.
7 days.
You know,
do you know anything about natural immunity?
Very little.
Okay.
Shane Crotty thinks that it could be durable, but we really don't know.
Will be highly unusual for a respiratory virus.
Yes, I understand.
Especially when there's no viremia.
Di writes, aren't these vaccines being distributed before the studies are peer-reviewed? They are.
Well, define peer review.
Yeah, you submit it to a journal.
What vaccine needs to go be a journal article?
Yeah. Really, the data just need to be looked at by the Data Safety and Monitoring Committee and eventually the FDA in the U.S.
Yes, and that is their peers. So technically, it's kind of it's peer reviewed.
Donald writes, researchers adapted WHO protocols for poliovirus population sewage surveillance to find SARS-CoV-2. That's right. That's right. Will we ever be able to monitor for novel viruses this way? How can we scale up novel sequencing? I think that's a great question. Can you find novel viruses that people might be shedding in sewage? And the problem is we don't really have a catalog of the human fecal virome, right?
Well, it's reflective of the gut virome, right?
It is, yeah. But
so you have people have some catalogs of the gut virome.
Yeah.
And you don't need to scale up next generation sequencing that much. I mean, it's very high throughput.
I've heard that COVID disease can be a result of either an insufficient or, I lost it.
Excessive T cell response. Is the insufficient response typical of older patients and the overactive younger? That's the thought. It hasn't really been proven.
You know, we are really early on in this. That's the problem. You have to wait 2 months after getting the vaccine and get tested for antibodies to make sure you are protected. So you're asking, if you get the vaccine, both shots, most of them need 2, can you just go out and not worry about getting infected? You know, the vaccine is not 100% effective. It's 90 or 95%. And out of millions of people vaccinated, that's a lot of people who are not going to be protected.
Yeah. I mean, the difference between 90 and 95 is twofold.
People get monoclonals as a preventive and then get exposed to virus. Wouldn't that form immunity because the body was involved in the fight? So if you get monoclonals, they'll last maybe a month. And the monoclonals will prevent you from getting infected for about a month. So if you are infected, yeah, you won't get your own immune response. That's right. So that's the thing. If you're treated with monoclonals, it really will prevent you from making your own long-term immune response.
I like this. Is it true that SARS-CoV-2 hijacks lysosomes and uses them as vesicles to leave the cell? Unfortunately, that paper was based on murine hepatitis virus slowly. And she did like one maybe pseudotype virus experiment with SARS-CoV-2. It was like totally disingenuous in the title. It was appalling.
So Ray wants to know, what made the speed of vaccine development possible? Investment in science for 30, 40 years, you know, development of all the basic methodology. For example, the ability to make a ton of mRNA, that was developed years ago. What's the difference between Pfizer and Moderna? So it's the same mRNA encoding the same protein. The lipid nanoparticle coating is different. And that's why the Pfizer one has to be kept at lower temperatures than the Mrna. But I think we don't know what the difference is because they don't tell us. It's a proprietary thing.
Has to be similar enough to fuse with the cell membranes. It has to be rather similar, more like 90%.
Any thoughts about the future regarding long-term COVID treatment? I have long COVID. That is a really good question. People are working on it. There are some ideas. If you listen to our TWIV on long-term COVID, you heard some of the people talking about it. But my understanding is there's research going on, and hopefully people will come up with something. It won't be ignored. It's very difficult to model in animals, absolutely. The J&J vaccine is a one-shot vaccine. That's correct. Whether or not, we'll see what the efficacy is for that. We just don't know. Oh, you did the lysosome one, right, Amy? Yeah. Why was the UK vaccine approval so fast compared to the FDA? Well, they do it a little differently in the UK. There, they don't actually have a board, an independent board that looks at the data. They get their data from the company, and they go by the company's word. Whereas here in the US, the council takes the data from the company and they look at it independently and make their own decision. So that's the difference, and that's why it could get approved so much quicker in the UK. Is Moderna a full spike? Yes, both Moderna and Pfizer are full spikes. Could the speed process of vaccine development for this virus inform and accelerate vaccine development for other less pressing illnesses? Sure. Especially the mRNA vaccines can be widely applicable to any other disease where you need to deliver a protein to people. But yes, the whole idea of doing it very quickly, overlapping phase 1, 2, and 3, and still being safe, I think is a lesson. Because we've never made a vaccine this quickly. No matter, we've made so many different vaccines, which is really remarkable when you think about it.
Right, it's 8:30. I gotta go put on my overall.
Bye, Amy. Okay, do you still have echo when I'm here alone? Let me know. Hey, Snowfox, really? You think I don't know what a podcast needs? I've been podcasting for 12 years. It was Amy with the echo, I think. Now tell me if you hear echo on me or not. Okay, so we're fine. It was Amy. I don't know what's the problem, but as I said, she's 40 miles away, and I couldn't help her. But I'll try and sort it out this week. Okay? Do you want to ask some more questions, or do you want to I'm willing to stay for a while if you want to ask some more questions and not talk about echo, okay? How long will it take to sort out the dosing issues for the Oxford vaccine? That's a great question. They're not being very forthcoming. And, we're hearing all sorts of things, and they're not really telling the public. They're telling their shareholders. So I don't know how long that's going to take. It's really unfortunate because that could be a good vaccine. It's inexpensive. It doesn't cost a lot of money. So I'm hoping we get that pretty soon. You guys are still talking about echo. vaccine trial for children. Do you think it's safe? So we had on Immune the other day an immunologist who's a pediatric immunologist, and she said there's no reason why we can't do vaccine trials in kids right away. It's safe. And, you know, to say you can't give the vaccine to under 18 right now because they haven't been tried yet isn't really fair. So I agree that we should be doing it in kids. The Oxford vaccine might be better for someone with an autoimmune disease. I don't know why that would be the case, Laura. It's not clear to me that makes any sense. Any progress on the home paper test? No, not that I've heard of. It's really kind of disappointing. I would have thought that it would be done by the end of this year, and I'm talking about Michael Mrna's approach. You know, I think we'd love to have a lick a stick, right? A buck a stick. You lick it in the morning and see if you're infected, but it's not here yet. I don't know. Maybe I've been talking to a lot of people about it, and it's going to happen. It's just not happening as fast as I want. Hey, big hello from Ireland. Has COVID ever been isolated? Well, COVID is the disease, right? SARS-CoV-2 is the virus, and the virus has been isolated thousands of times from many different patients. So that is not an issue. Should long COVID patients be concerned about taking the vaccine? This is a good question. I think until we have some more information on what's going on in those patients, we have to be careful. You know, it's a particular risk group of patients, and certainly the vaccine's not been tested in them because they have been infected. So I think that's an important issue. Do you think variability in the virus will cause trouble in the vaccine? No, I don't think so. You know, none of the coronaviruses vary enough to evade immune responses. It's just not a coronavirus thing. You know, it's a thing with influenza viruses. There's variation in the virus and they can evade vaccines, but it's not a coronavirus thing. Of course, there's always a first time, but I don't see any evidence of it. I don't see any change so far in all the isolates since January to now where that would evade vaccines. How long can SARS-CoV-2 survive on surfaces such as skin or clothing? It can retain infectivity for many hours, probably a day. Whether it can be transmitted that way is a good question. There are no good epidemiological studies that suggest that. Nevertheless, it's good to be careful. What are the important lessons of this pandemic? I'll get to ivermectin in a minute. Hang on. Okay, there are, and the mink strains too, I'll get it. The most important lessons are, first, we weren't ready for this one. We could have been. I've said this many times, and many of you have not listened to me before, but we should have been ready for this. We could have been. After SARS-1, we could have had antivirals and maybe broadly reacting vaccines ready. And this should have never happened. This whole nonsense should never have happened. So I think one of the lessons is next time we should be ready for the vaccine. The other lesson, of course, is we have to have a better preparedness plan. You know, we didn't have any plans for PPE, for hospital space, and for managing this. Every state was on their own. So I'm hoping we learn, but I'm not sure. Are monoclonal treatments available to the public yet? They are under an emergency use authorization. You have to be in certain risk groups. So you have to have a positive test and be in a certain risk group. For example, have other diseases that are comorbidities, and then you get it by prescription. You have to go to an infusion center and get it, of course, because it's intravenously delivered. Ivermectin, I still have not seen convincing evidence that ivermectin can impact clinical disease. I know it's a great drug for other things. I have not seen it for ivermectin. What is it, the other one? Oh, the minks, yes. The minks. So the mink situation seems to have calmed down a bit in Denmark. This one strain that arose in mink that had a number of changes that seemed to make it less well neutralized by human serum, that seems to have disappeared. And I think the culling has really helped. And my take on this is if they carry out biosecurity on the farms, in other words, have all the workers tested before going in on a regular basis and do good isolation, that they can prevent more outbreaks like that. Vitamin D, go for it. Vitamin D, I started taking vitamin D months ago because I'm sitting inside most of the time. I'm not getting any sun. And a doctor who I know well, she said, everyone should take it and take it in reasonable amounts. And there's some data that suggests, it could modify the severity of COVID-19, so why not take it? I didn't see Paul Offit's JAMA interview. I'll take a look at that. We can talk about it next time. Goodbye, those are leaving. Will I be interviewing anyone on Biden's coronavirus team? I'd like to. We have enough of a profile that people come on when we ask them, so we'll see what we can do. So yeah, there was a SARS-1 vaccine that was never completed. But I'm not sure that would provide cross-reactive protection. We never checked it, but it was a proof of principle. Yeah, how many people need to be immune? If the reproductive index for this virus is 2 to 3, then it's 50 to 70% of the population has to be immune. And I think 70% is a reasonable number. So if we can get 70% immune, you'll start to see dramatic decreases in transmission. I had Sabin polio vaccine. Any good against COVID? Not if you had it more than a year ago. So, there are a number of reports that say that these attenuated infectious vaccines like the Sabin vaccine, MMR, the tuberculosis vaccine, give you some protection against SARS-CoV-2 infection for a few months because they kind of generally boost your innate responses. So if you get such a vaccine recently, that's great, but not 10 years ago. About the outbreak on the Oregon mink farm. So all these outbreaks are a concern because they establish a new reservoir for the virus, right? And you don't want that because you make a new reservoir and it will keep infecting people. Plus, it could change in the animals in a way that makes it harder for us to deal with. So it's not good whenever there are large numbers of animals infected outside of humans. Any reason to prefer Mrna over Pfizer? I don't see any reason. I think the data they both look good. And, the only difference is the Pfizer needs to be kept frozen longer. We had Shingrix dose 2, that's good, but that's an inactivated vaccine, so it doesn't have the same considerations. What percent of the population has been exposed? If you do serological surveys, it really varies depending on the population. There is, a study out of New York City in a neighborhood called Corona, by the way. It's in Queens, where a huge percentage of the people are seropositive. In other areas, it's less than 1%. I didn't interview Michael Osterholm. I wanted to, but he doesn't like me, so he doesn't want to come on TWIV. He disagreed with me several years ago, so he hasn't been coming on. What advice to someone who just got diagnosed with COVID? Well, if you are, the real issue is if you start to have shortness of breath, right, then you have to be hospitalized. So, don't overexert yourself. I know Ian Lipkin, if you remember, some of you may remember, came on TWIV after he contracted COVID, and he was doing too much, and he found himself getting short of breath, so he just took it easy. So make sure you're monitored frequently by a physician to make sure that there's no issue. I mean, there's not much you can do at an early stage until you have shortness of breath. Now, if you do have comorbidities, a physician will possibly get you monoclonal antibodies if it's early enough in the disease. We disagreed about, Hosterholm and I disagreed about, all right, so avian influenza, H5n1, that was a big deal years ago. Nobody's thinking about it now. We covered it a lot on TWIV. So these are influenza viruses that infect birds, chickens, and ducks and geese, and they kill them. And occasionally they jump into people, and they can be lethal. And I argued that there are many asymptomatic infections with those viruses, so it wasn't as lethal as people thought, and Osterholm disagreed with me. Why do some people have asymptomatic responses? Does it correlate with the strength and length of the innate immune response? It may very well be. In fact, I just saw a paper where it suggests that we may do that on Friday's TWIV. Amy's going to be on Friday's TWIV. And it suggests that maybe people with asymptomatic infections make a better innate immune response initially, and they can just contain the infection. And even though the virus is still reproducing, it doesn't make you sick. Oh, this is a good one. Does SARS have any genes that affect one's interferon response? Yeah, we actually just talked about that on today's TWIV. One of the viral genes encodes a protease, right? It's an enzyme that chops up proteins, and that can dampen the innate response. It's very cool. Any evidence of human to wild animal transmission? So Tony Schounts at Colorado State is checking that out. He's going to look to see if deer mice are getting infected from humans. But someone must be looking at bats. I'm sure they're doing it as we speak. How does SARS-CoV-2 affect the immune system? Well, it makes it go crazy. All of these post-infection symptoms, right? So you have this first viral infection, you have an upper tract infection, you may have some lung issue, and then for weeks you may have multi-inflammatory syndrome. You can have kidney issues, gut issues, skin issues, neurological issues, and those seem to be driven by an aberrant immune response. So that's what I would say to that question. Why does COVID affect people of color more? Well, lots of reasons. We had a wonderful guy on TWIV a while ago who talked about that, because they don't have access to good health care. They generally have more comorbidities, and they're often at the front lines working, and they get more infected more readily than others. Are HIV protease inhibitors still an option against SARS-CoV-2? No, they don't. The clinical trials I've seen, they haven't worked. I'm sorry about that. A number of people have asked how long the nanoparticles stay in the body. So some people are concerned about long-term issues, but these are not like metal nanoparticles that you're thinking of. We can manufacture really small metal nanoparticles. We use them for many things, and they can persist. But these are biological products. They're made of lipids, and they do not stay around forever. They are rapidly taken up by cells. And I would be very surprised if these particles stayed around more than a couple of weeks. So there you go. Hey, today we have 100,000 hospitalized people with COVID-19 in the U.S. That's a record, of course, and that's twice as many in the spring. So a lot of people are getting very sick these days. Any updates on ISG SNPs and SARS-CoV-2 infection? Not since that one paper that we did a while ago came out in Science. I haven't seen anything new yet. Has an mRNA platform been considered with RNA interference? Yes, but you know what the problem is? It would take longer to develop. They needed to do vaccines that could be developed very quickly, and mRNA is one of them. Adenovirus is another, because it's a known platform, and that's really what drove the current development. Why would Amy want to hold out for an attenuated virus vaccine? Because the attenuated virus vaccine mimics the real infection, right? You take a virus that actually reproduces in you. It just doesn't make you sick. And the Sabin polio vaccine is an example. The measles, mumps, rubella vaccines, they're all like that. So they're very effective. And she feels that that would most closely mimic a natural infection. But we don't have any of those being developed in the U.S. As far as I know, there's just one. There are a few in other countries. Any new evidence on whether the infecting dose could have an impact on disease severity? I have not seen any good data. I really do look carefully at the literature, and I haven't seen anything which suggests that the more you get, the more severe the disease is. Now, the more dose of virus you get, the more likely you are to be infected, right? So that's something. If you have just a few particles, you wouldn't get infected. So that's where it is, but not in terms of disease severity. I heard SARS-CoV-2 has a low mutation rate because it has a proofreading enzyme, which is unique to coronas. Yes. So RNA viruses all make a lot of mutations, and they can't correct them. They don't have a correcting enzyme. The coronaviruses have a huge genome, 30,000 bases. And to get that big, they have to have a correcting enzyme. So the error rate is lower than other RNA viruses. And that's why it's hard to make antivirals that target RNA synthesis in those viruses. Why is Amy so argumentative with the other members on TWIV? Well, she's, listen, I've worked with Amy for 17 years, and that's just who she is. She's very smart, and she doesn't tolerate when people say dumb things. Okay, can I put it that way? I don't blame her, and I've learned to deal with it. But other people don't often do it. I think she's terrific. Yes, she's really someone to appreciate. Her mind is amazing. That's why I have her on this, and I'm going to fix her sound because having her with me really would make this a great Q&A every week. And I'm glad she's doing it. She went back to do experiments. It's 8:30. She's got to do experiments now in the lab. She's really dedicated. Yeah, so Stella writes, I sort of feel like the focus has shifted from therapeutics to vaccines. I think that's the public focus, right? Which is driven by the press. And I think behind the scenes, a lot is still going on with therapeutics. A lot of antivirals are being developed. For example, I know at Columbia that many people are working on antiviral approaches, and we need these for the future, right? We need them in case there's an outbreak in unvaccinated people. So you're going to see more of that. It's just that right now we need vaccines to get this over with because the world is being trashed by this, right? And we have to prevent people from getting infected. Who are excluded from vaccine trials? Anybody under 18. The first set of trials, 18 and up, and then shown to be safe in them, then they will move down. That's the traditional way of doing it. Now, so yes, some doctors are saying that PCR is not good. It's not useful, but that's just simply not true. PCR is very useful. The only problem I see with PCR is it's too sensitive. So you can get a positive test like I did and not really be infected. It could be a false positive, or it could be you have very low RNA, so you're not transmitting, in which case you're not dangerous. That's the only drawback. And that's why we need the rapid antigen tests on a stick. How can we trust mRNA vaccines when the phase 3 trials are so short? We don't know if they have any long-term effects. Okay, that's a good question. And I don't blame you for worrying, because this is the fastest vaccine development that's ever happened, right? But I'm looking at the phase 1, the phase 2, and the phase 3 trials. And all along the way, even though we're talking about less than 100,000 people, I haven't seen any serious side effects. But that's why the next month is important, because the most serious one that I'm worried about is Guillain-Barré, which can happen in the first 2 months after immunization. And if we can get past that, then I'm confident. And then you make a risk assessment. You have these safe vaccines. You've only been tested for less than a year in so many people, or do you want to worry about getting serious COVID. That's the choice you have to make. And I think the data are pretty clear on that. Are there epitopes on the spike that could produce autoantibodies? We haven't found any, but it's possible. And many virus infections trigger autoantibodies, not by mimicry, but by exposing auto antigens in cells. So you can't rule that out. How do we achieve trials with children since they can't provide informed consent? Well, their parents can. I've seen the consent forms, and the parent signs on behalf of the child. If you're a minor, the parent can do that. How important are the CT values for treating COVID patients? Well, so treatment really is only when you have trouble breathing, right? And so by that point, knowing the CT value is not going to help because you're actually beyond the virus phase of the disease. Where CT is important, I think, is telling you if you can transmit or not. If the CT value is 35 and above, you're very unlikely to transmit. But the FDA in the US doesn't give out CT values, so it's of limited use. Have insects been ruled out as carriers? I have seen a few papers. No evidence, as far as I can see, that the virus reproduces in insects or that they can carry them. I think that's not an issue. Vaccinating people who have already had COVID, helpful, useless, harmful? I don't think it's a problem. I see no reason why it would be an issue. I don't think you're going to be stratified based on whether you had COVID and not. Is there any reason not to get all the different types of vaccine? They're not being tested that way. So I would say you should take them in the manner that they've been tested. You get an mRNA vaccine, you get 2 doses of the mRNA vaccine and not an mRNA and an adenovirus vaccine because you don't know if that's going to work. Yeah, prisons are actually one of the first targets for vaccination because they're closed populations, and also the people who care for prisoners are at risk as well. Has any mRNA previously been tested on children? Not that I know of, no. It's only been in adults. Why do some people develop acute respiratory distress and not others? I think the emerging idea is that the virus gets out of control early on and reproduces to very high levels, and that overstimulates the immune system, and then that causes the damage. And so the earliest times are important. I think the innate immune response is a key player there. I think the virus has the ability to overcome the innate immune response, and those people who are better at countering are the ones that do well. And that is that's going to be tested in the next year or so. Would I take a vaccine? Yeah, I would, but I don't need to right now. I want to step aside and let other people have it who need it. I would take these mRNA vaccines for sure. I think they're quite remarkable. But I mostly stay home and go to my lab once or twice a week, and I feel like I'm at low risk. And if you want my dose, you can have it. Not because I don't think it's safe, but I just want you to have it. What happens if you get Pfizer for 1 shot and Moderna for shot 2? I think that's okay because they're basically, the mRNA is the same in both, right? So I don't see why that would be an issue. So there is an advisory committee which is suggesting to the CDC who should get vaccinated first. And, first are healthcare workers and that sort of, then very seriously ill patients and elderly patients and so forth. And then essential employees, maybe they were number 2. Who's going to decide? The CDC will decide basically who's going to get it first. Would you have to keep track of which type of vaccine you've been getting? Well, whoever gives you the vaccine will certainly do that. You'll get some kind of a record, or they'll keep a record of it for sure to know what you've had. Is there any risk assessment for which individual risks are clearly outweighed by evidence? No, nobody's done that, really. I think that's an interesting point. But But right now, they're just trying to get something out there that looks like it has high efficacy, over 90%, and curtail the spread of virus, because right now it's horrible, right? Especially in the U.S. Would getting an adeno vaccine make any future adeno vaccines useless? Great question. So you noticed in the AstraZeneca vaccine that if they gave half a dose for the first dose and full dose for the second, it worked better. And that may be because there was less antibody made against the vector. So yeah, if you get a chimpanzee AD26 vector, which is what the AstraZeneca is, in the future, using the same vector in you is going to be a problem. So you would have to get a different one. All right, we got 2 minutes left. And let's do a couple more questions here. And I'll be back next week at the same time, and I'll fix Amy's sound, all right? I promise. I promise. You see, my sound is good, but I was really stumped. Is there a possibility that the spike that is used in the vaccine occurs anywhere else? The whole spike, no. I mean, small bits of it may be related elsewhere in the body. I haven't seen any evidence to that so far, which could, of course, be an issue. You would get autoimmune reactions, but so far we haven't seen that. So someone's asking, what if we don't get to 70%, but we get 50% uptake? Well, that's going to make a dent on transmission. It may not eliminate it entirely. I think what's going to happen is as the vaccines are distributed and people see that they're okay, they're safe, more and more people will take them and we'll get to 70%. That's not a hard number to reach. In the U.S., we do pretty well. We do over 90% for many vaccines. Hey, Stella, I'll see you tomorrow night, right? I'm doing a UW-Madison thing online. That'll be cool. You guys like this? Was it useful? I'm sorry about the Amy thing. I'm going to try and sort it out this week so we can do this again. Okay, folks. I appreciate your coming. See you next time.